Today we are going to learn about genome variation or genetic variation So all of you know that the gene is the fundamental physical and functional unit of heredity, and it is composed of nucleotides like gene composed of nucleotides, and then after that, like, it gets transcribed to mRNA and gets translated to protein. So in your experiment 1 you have learned about the sequence variation of actin gene in different crop species. So today you are going to learn about the genes which have variation among individuals of the same species. So there are three kind of variations we are going to study. One is SNP or single nucleotide polymorphism, and two is insertion and deletion. There we can call them indels. And then third is transposable elements or transposons. So the first one we are going to do SNP or the single nucleotide polymorphism. We can call that as a SNP. So for example, in humans, sickle cell anemia, is due to a single amino acid change or due to a single base pair change in the beta globin gene. In that, GAG codes for glutamic acid. This is a normal condition, and it gets changed to GTG. It codes for valine. So the change from glutamic acid to valine results in the sickle cell anemia. So it is, that's why single nucleotide polymorphism. So the polymorphism is, and this A turning into T. Okay, and then second is your insertion or deletion. So this is one or more nucleotide either added, like the addition, or removed or deletion, results in the frame shift mutation. If it is one or two nucleotide, definitely it will result in a frame shift mutation that results in, like, earlier termination of the protein. So as a result you will not get the active protein. For example, we can TTA, GTC, ATG, and then TTA. For example, if For example, if A is added, one nucleotide, so you will have GTA ATT. So when it gets translated, transcribed and translated, it will turn in, for example, here T, the other strand. So finally you will get into, when this gets transcribed, you will have UAA. So you will have a stop codon. Same way, if another A is added also, definitely another, for example, no sorry, T is added, then again it will end up prior termination, instead of like now it's going to be ending here. So okay, the third part is transposable elements. So this we are interested in because Dr. Wessler lab for the past 3 decades she's working on transposable element both in maize and rice. So the transposable element, there are 2 kinds. One is, you can say, autonomous, and then B is non-autonomous. So autonomous elements have the characteristic gene that codes for transposes, and then they will have, okay, I'll put this. They will have characteristic terminal inverted repeats, and these repeats are more than, like now usually more than 16 bases long. So for example, what is terminal inverted repeat? Say example ATGC DACG. And then this I will put 1, 2 sequence, and then here it will be ATGC DACG. This will be 3, this will be 4. So 1 and 4 are terminal part that is inverted there. Same way 2 and 3, they are inverted there. So TACG, you can see TACG. So this is characteristic for the autonomous element. So the non-autonomous element, it will not have no transposes. So this non-autonomous element won't be able to move by itself. So the presence of single active autonomous element will help in the movement of its own as well as the non-autonomous elements around the genome. So in this experiment we are going to use maize as a experimental model because it's well studied.